Thanks a lot, Tina, for this very nice introduction. And thanks a lot, the EPNOE community and the organizers, that I have the chance to be present here at this webinar and show you a few of our ideas that we have, and also about our plans that we have regarding polysaccharide peptide conjugates. So you might ask now, what are polysaccharide peptide conjugates, for those of you at least that are not so much into that topic, and that's why I'm also going to share with you a few slides about the introduction, about the concept of it. So why we like to conjugate these two, say, biomolecular building blocks. So that's why it's termed why and how, and I will try to show you a bit about it. So for myself, I'm, as Tina said, Rupert Kargl from Graz University of Technology, and as Tina already explained, I have a bit of a background in chemical modification of polysaccharides, and I'll try to switch to the next slide. Okay, so just for those of you who have never heard about it, about our relatively new institute, I just would like to introduce a bit what we are doing, because we are in a way a merger between also two worlds. So it's not peptides and polysaccharides, but it's polysaccharides and monosaccharides or oligosaccharides. And you know that in glycobiology, there is a lot going on, of course, in derivatizing small building blocks of polysaccharides. And the vice head of our institute, Professor Tanja Wodnik, and her group, so the so-called Glyco group, they are very much experts into that. And you can see here, and I'll try to move with a pointer, it's maybe more instructive. You can see here on the bottom some examples of some structures that this group is producing. And there was also an introductory talk in one of the webinar sessions by Professor Wodnik about this. And the other part is where I'm working on a bit more, is the technology part, and you can see that it's applied material science in a way. So we try to kind of process chemically modified, also non-modified polysaccharides mostly. And that's where the idea of combining peptides and proteins with the polysaccharide materials comes into play. So just to give you a very quick outline about the, say, work processes that we have at our institute. Of course, as many of you as well, we start with extracting polysaccharides from natural resources. This is, I would say, not our main core expertise, but we usually purchase them, of course. What you can see here is extracted alginate, right? So, of course, alginate is one of the very interesting materials that we work with, and it's quite abundant. And then we usually go on and do some kind of chemical derivatization, because obviously we're not super happy with the, say, naturally occurring properties that these materials have. So we do some kind of technology and attach, usually very often, something to the backbone of these materials. And then we do analysis, and this is somehow symbolic. There's a symbolic representation of the analysis done here. So of course, it's either some interaction with mass, right? It's gravity, gravimetric analysis that you do, or the other option is, in any kind of analytical method, you have an interaction with an electromagnetic field, be it NMR or be it mass spectrometry. It's always some kind of changing electromagnetic field that gives you some idea about the chemical structure of the derivatives that we produced. And then, of course, the processing part can be, and this is also symbolic, just symbolic here, can be, for instance, 3D printing, but it doesn't have to be. But since 3D printing is very fashionable nowadays, we also, of course, have one or a few 3D printers in our labs. And the example that is shown here is a printer that can mostly do extrusion printing. So you have some kind of cartridge and you extrude some kind of flowable material out of these cartridges and shape it into certain forms. And the main idea or the main aim of our institute is also, or mostly, I would say, to apply these materials in biomedical fields, because this is where we think it has the highest potential. Because of course, in the more technical parts, we are competing a lot with synthetic polymers, but those are of course not that complex in terms of chemical structure. So we think that in biomedicine, our structures are quite useful. That's why we mostly go into that direction. And then the question comes up, so why PPC? So polysaccharide peptide conjugates. This was the abbreviation that I termed at the very beginning. So it will be a part of this presentation as well. So you will see why PPC. And I would like to just show you two things today. Of course, there are different motivations. It can also be just a scientific curiosity why you would like to do such things. But we have been, and we're still motivated by the fact that there are two different distinct structures that are already existing in natural systems, so in living systems. If you look very close and if you take a look at the literature in biology, you will see that one example are so-called proteoglycans. And of course, the name already tells you that it's a combination of proteins and a glycan. So there is a lot of literature about it and studies. And a representative example of one of these proteoglycans is a combination of the very famous hyaluronic acid, which is depicted here on the bottom. So the simplified structure, so to speak, you know that it's an N-acetylated glucosamine, and it has some glucuronic acid in it. So it's a copolymer, and it has some certain functional groups, of course, and it's found in all of our tissue. And these are non-covalent, say, complexes that are formed in biological systems between the backbone of this hyaluronic acid and some kind of branches that you can observe, which are polypeptide chains that are depicted here in the form of the black lines. And what is observed in nature, or what people found out some time ago using all these nice analytical tools, is that you have some interactions with the nitrogen end of this polypeptide chain and the hyaluronic acid. And these interactions is non-covalent usually, so it's some kind of folded peptide that arranges around the hyaluronic acid. So this was this is one of our motivations to create this kind of PPCs, not really from scratch, but from building blocks, as I said. So just remember that these extracellular proteoglycans, they bind hyaluronic acid and form these large complexes. And moreover, these polypeptide chains, but this is not a part of the content of the talk of today, but just to mention, these polypeptide chains, they are of course also glycosylated to a certain extent, which makes them very hydrophilic and causes the high viscosity and water binding capacity of these kind of structures. So just to have in mind, proteoglycans is one of the motivation for our work. Then there's another motivation, which is kind of in a very different biological organism or organisms, and these are peptidoglycans. So it's again, as the name says, of course, a combination of a peptide and a glycan. And in this case, as you know, the fraction of the protein fraction of the whole structure in this cell wall of bacteria is much lower than in the proteoglycans. That's also why they're called peptidoglycans, right? But what it essentially is, at least what the scientific community thinks it is, it's kind of a chitin backbone, if you wish. So you can think about it like a chitin backbone, so N-acetylated glucosamine, beta-1,4 linked. But every second unit, as you can see here in this picture from this nice review, every second unit has on the, say, second hydroxyl, something like a lactic acid ether bound to it. So it's again a copolymer, and this lactic acid ether makes it possible that there are also peptides attached to the backbone of the polysaccharides. And usually these are something like 5 or 6 or residues that link the adjacent chains together and make a very strong network. This is what bacteria are capable of doing, of course. And you can see here on the left an electron microscope image of such a peptidoglycan. It's called saccharose, as it is mentioned here in this review. So it's a very thin layer if you dry it out, of course, in the nanometric range thickness and in the size of the bacteria itself. So of course it protects the bacteria from exploding against the osmotic pressure and holds them together. And as you know, it's also a target of many lactam antibiotics that interfere in the biosynthesis of these kind of peptidoglycans. So just to remember for you, the second motivation for us is somehow to be inspired at least by these natural structures of the peptidoglycans that are of course super complex and cannot be made chemically that easily. Then I have to say that of course there's a lot of research and interest in polysaccharide protein or polysaccharide peptide conjugates in general going on. So this is also the reason why several groups have published reviews about this topic. So it's of course not only about this peptidoglycans and about the proteoglycans, but also many other different applications. So research groups are interested in this kind of topics. And I will explain what their interest is and also our interest partially, and for what purpose this PPCs have been used so far, at least in research. And as you can see here, so there are two examples maybe of the latest reviews that people published, one in carbohydrate polymers and the other one in advanced functional materials, and both of them cover similar topics. So how to chemically produce PPCs for the one or the other purpose. So if you're interested in that, of course, these are very nice reviews. And we also contributed a bit to that topic. So some time ago now, we also published a review covering a similar topic, but maybe focusing more on PPCs of three distinct polysaccharides, namely dextrane, chitosan, and alginate. And the reason for it is because we thought that the reactive groups that are in these polysaccharides, or the functional groups that are in these polysaccharides, so the carboxyl group, the amino group, and of course the hydroxyl group, they are somewhat representative, or I wouldn't say all, but many of the existing polysaccharides. So this is why we've chosen these. And in this review, if you're interested, you will find it. We also listed on the one hand, and that's what you can see here on the left, what people did or published before these years on different polysaccharides. So you can see it's kind of a collection of articles or statistics covering very different polysaccharides, so from chitosan to chondroitin sulfate, in combination with some kind of peptides or proteins in terms of conjugating it chemically. And you can see that there is obviously a certain, say, trend. So something happened in 2005 that caused more papers to be published on that kind of topic. But of course, if you look very careful into the history of this kind of research, you will also see that already in the 1960s, people tried to conjugate first of all amino acids to polysaccharides, and then later on larger peptide structures. And the main motivation at the very beginning was to do affinity chromatography. So it's, the Cephatel's column chromatography. This was the main motivation at the very beginning. And later on, you will see, if you also look into such things, many different other potential applications came up. And these applications, they are also listed here. So of course, this is not a complete list. That's why there are some dots. But you will see that if you look into literature about PPCs, a very big topic is immunology. So there are even vaccines produced that combine polysaccharides and proteins. These are commercial vaccines because immune cells respond to it. A lot of topics are tissue engineering. Because of course, if you have a gel of a polysaccharide that is combined with a peptide, that can be chemically or enzymatically cross-linked, or gels You can do something like tissue engineering in conjunction with proteoglycans. Of course, the rationale is that you imitate the extracellular matrix. There is a lot on drug delivery. So colleagues of us also published, for instance, DNA delivery into cells using at least an amino acid polysaccharide conjugate, complexing the DNA with it and delivering it to cells. And you will also see that there is a lot on imaging and sensing. And maybe just the sensing, because it's more exotic a little bit. I put here an example of a group. So this is of course not our work, but it's cited here of what can be done in terms of sensing. So this group in a paper published just recently conjugated fluorescent dye peptide construct to the backbone of dextrane, as you can see here. And this dye that is coupled is only fluorescent once the peptide is cleaved by an enzyme. So with that quite smart construct, you can make responsive materials based on the backbone of a polysaccharide. Of course, one has to mention that the backbone itself doesn't have the biggest role here. So the function, of course, comes from the peptide and the dye, but of course, there's certain biocompatibility of the backbone. But just as an example, what can be done with sensors and smart materials. And then, of course, the question comes up, and that's maybe the core topic of today's webinar, is how to make these PPCs. And I would just like to list you a lot of different possibilities, and this is just a quick overview of what I think is the essence of it. So you have on the one hand, of course, your polysaccharide, however it may look like, but it will always, at least this is what we know so far, right, have something like a reducing end, and this will be mostly an aldehyde in a natural state or even an extracted state. And you will, of course, have a non-reducing end, which will mostly be, not always, but I would say mostly be a hydroxyl moiety. And of course, every polysaccharide in between has a lot of different functional groups that I mentioned already, but still you can group them into the most common ones and the chemically most similar ones. Of course, the hydroxyls, and you will have, as we have heard about before already, carboxyls in many cases, like in alginate, and you will have, for instance, as in chitosan, of course, amines, and you will have something like acetylated amines as in chitin, and so on and so forth. But I would say that those are maybe the major ones in the backbone. And of course, what is maybe overlooked a lot in some cases is you will always have water in one or the other form on the backbone, even in an organic solvent, right? It will never be completely dry, I assume. But of course, you could also then have on the polysaccharides some minor functional groups. So aldehydes I mentioned, of course, in the reducing end. But this can also be due to, say, chemical oxidation that happens in the chain, or also to chemical oxidation on demand. So a lot of people try to oxidize hydroxyls in the chain, of course, to produce the aldehydes. And you will have possibly sulfates and phosphates in the polysaccharide if it's especially coming from natural systems where proteoglycans play a role. And what is maybe the least common, I would say, is alkenes, but even those, if you think about this oxidative cleavage of non-reducing ends, you can have the alkenes present. Ketones are also, of course, one part. And then I don't have to tell you, you know all these things, but just to repeat a bit, in the case of a peptide, you will have, say, nitrogen end and a carboxyl end, as far as we know, usually. And the peptide itself, depending, of course, how it is composed and how long it is, can be comprising also the very many different molecules. And I would say maybe sulfur is the one, or sulfhydryl groups are the ones that distinguish it a lot in terms of functional groups from the polysaccharides, because those don't have that much sulfur present usually. And then, of course, you will have a lot of hydrophobic groups that cause the association. And of course, you can also have glycosylations, just to summarize. So you can imagine, you know, it's a very complex thing to teeter those two things together. But to make the story short here, what is shown here on the right side are 4 examples of what people did trying to conjugate the chain. Mostly the chains, I'm not talking about the reducing end now, the chain of polysaccharides with peptides. And of course, if you have carboxyls and hydroxyls, you make nice esters, if you're lucky. So that's an example of dextrane ester, in this case with an amino acid, protected one. Or if you have alginate, people did, of course, a lot of amide chemistry to bind this peptide from the other side, so from the nitrogen residue or end of the peptide chain. And then there are the more exotic ones, or the more, say, chemical ones, because there is a lot of work on spaces, where, for instance, chitosan in this case was also modified in the form of an amide. And then the very famous and Nobel Prize winning click chemistry was used to teeter peptide to the backbone of the chitosan. And of course, you have to have then modified peptides that are usually provided by chemical synthesis on solid phases that are equipped with these reactive functional groups that can form this backbone. And the last example here I put because this is also one of the most common ways of putting linking peptides and polysaccharides in literature. It's oxidation, as I said, so it's introduction of aldehyde groups, usually by oxidation, and then reductive amination of the present amine groups of the peptide. But as you can imagine, this, of course, is not very specific to the nitrogen end of the peptide. So as long as you do not have protection groups along the peptide structure, it's quite clear that you will not be able to control very well at which position of nitrogen the peptide attaches to the backbone of your polysaccharide chain. So I'd say that in very quick, a few minutes, this is maybe the essence of the coupling chemistry. Of course, there are a lot of different others, but in most cases, I'd say that coupling agents are used to covalently immobilize the peptide on the chain. I would also like to point out, because I talked a lot about the in-chain modification of the polysaccharides, I would like to point out that there is a lot of work, very interesting work on modifying the reducing end of the polysaccharide chain of very different polysaccharides. And one early example of this is very nice work by French researchers who produced block copolymers of polypeptide chain and the polysaccharide, in this case dextrane, by also using click chemistry. And what was observed was that these block copolymer structures form vesicles. And I also would like to point out that, and that's quite far off today's talk, but just that you heard about it, that there is, of course, work where people also used enzymes to teeter these two things together, because they are more specific and they may be also more efficient, so they might not cause too many side reactions and problems of purification. So this is also part of PPC's stories, of course. And of course, also of EPNOE colleagues, there is also modification, chemical modification of cellulose in the same or colloidal state, just that you know. And people also try to teeter, say, two polysaccharides together, which is shown here. And I think a very interesting work, which was lately published, is also the non-reducing end modification of a polysaccharide with at least amino acids, what has been shown recently by Professor Kevin Edgar's group, with a very nice approach, I would say, of modifying the non-reducing end Okay, that should be on another slide. Good. So to just give you some examples of what we did so far, even though this is already some time ago, but to maybe show you also what is a bit tricky about chemically modifying it and also purifying the products and analyzing them. So I'm talking about in-chain modifications. So some time ago, we tried to chemically derivatize dextrane again with a protected amino acid using a coupling agent. So I mentioned, I would say in literature, in 80% of the cases, coupling agents are needed or used to immobilize peptides or amino acids, the backbone. And we did similar things. So of course, the very famous coupling agent and very reactive one is carbonyl dimidazole. I have to say from my personal experience that one has to be careful with the CDI in a sense that it's often observed that this is hard to remove from the reaction products. It might also, of course, produce carbamates on the backbone without reacting with the amino acid before. So just if you work with it, take a look into if there isn't something attaching, something of the CDI attaching to the backbone by itself. But what you can see here is so you can produce non-regioselective modified dextrane derivatives. And you can, of course, do the analysis and the workup with it. And just what I would like to point out, and I think I mentioned it several times in other presentations, but just as a repetition, maybe for those who do separation, what you can see here on the bottom right is an example of the mixture of the reaction product, which has in the GPC 3 different peaks that you can see here. And after purification, at least the main peak is the polysaccharide, the modified polysaccharide that you can see here. But still, even though we put a lot of effort into trying to removing the monomer from the polymer, the message is it's quite tough to purify finally. So if someone is looking into biological properties, I think one has to be really careful and maybe also devise new methods to separate products and reactants, because this polymer analogous reactions can be, of course, quite tricky. And what you can see here on the left is also some determination of the molecular mass of the products. So you can see we used here, of course, static light scattering after trying to separate the products from the monomers, and also UV detection, because of course the trityl product that is attached that you can see here will be UV active. So you can see the difference between the non-UV active dextrane, which is this baseline here, and the UV active derivative, which are these two curves here, and the others are the light scattering signals. But also here, I would like to, and this is maybe just my personal opinion, but for those who work a lot with polymers, and especially polysaccharides, I'd like to point out that within or below this curve of the elution volume, of course, you have your molecular structures. And since size exclusion chromatography separates by hydrodynamic radius, I suggest to be very careful to interpret what is measured below this curve at a certain point, because the separation efficiency might not always be as you would like to have it, especially when the molecular masses and the distribution of the substituents are very, say, different in one product. And another remark to the analytics. So we did, of course, NMR, which is necessary to publish stuff, right? So what you can see is, in most of the cases, of course, a shift from the 1 to the 1' anomeric carbon, which tells you that the substituent is attached, at least on the carbon 2 position. And you can also somehow find out that it's, of course, as I said, non-regioselective. So very tricky to get a super defined structure. But what is also visible here is, of course, the the shift of the free acid of the non-substituted monomer, substituted monomer, and the ester that is formed on the backbone. So this can be used to identify or clarify the reaction, success of the reaction. Somehow I would like to remove this because the titles are missing. Okay. There is another example, and this is also a bit of an old story, and I've shown this several times, but I put it here for a different reason in a way. So you can attach small molecules which somehow at least imitate, for instance, if you think about phenylalanine. And I put this here just to point out a bit that upon modification with such a small molecule that is simple in a way compared to a peptide that contains a lot of phenylalanine residues, we already observe, of course, something like aggregation just in water. So the, say, aromatic residues of this small substituent will already cause certain aggregation, which you can see by just doing very simple tests of incubating surfaces of these solid surfaces with these still water-soluble materials. So you will get, of course, a very different colloidal behavior and solution behavior upon modification. And that's also what we observed here. And as soon as you attach a larger peptide, because of course peptides are not all water-soluble, this aggregation will be much stronger. So that's maybe something that has to be looked in a lot if someone is interested in the colloidal structure of the materials that are produced, just as another message. And what is shown here is, again, NMR data. And that's another message regarding PPC production and coupling agents. The main point here is, in this a bit crowded structure, what has been used here is a coupling agent. And I mentioned now the third time that coupling agents are maybe the most common things that are used to produce PPCs. And what we observed is it's also, again, really hard to, if you have carboxylic residues on the polymer backbone, it's quite hard to remove or to avoid side reactions of the coupling agent with the carboxylate on the backbone. And this is visible here by this many small peaks of the coupling agent that obviously, as it is known for EDC, reacted to form some form of urea derivative on the polymer. Nevertheless, you can confirm that you produced or you coupled your functional molecule by also, for instance, charge titration that is shown here, because of course, if carboxylic acids are derivatized, the charge of the overall product is reduced. And this is what you see here. So the starting charge of the polysaccharide is 3 millimoles and the end charge is something like half of it. So it's a nice method also to analyze. So I mentioned colloidal properties. This is just another example of the same chemistry, more or less, but it goes more into the direction of coupling, future coupling peptides. This is of course not a peptide, but it's an amino acid. And what I want to say here is what we did was we used methyl ester protected amino acid. So the intention was to couple the nitrogens or amine group alpha in alpha position to the backbone of the polysaccharide. And if you now remember at the very beginning, I said that in proteoglycans, the nitrogen end is bound to the uronic acid. And that's also, in a way, if you want, the start of the proteoglycan structure here. So we try to make amide bonds with coupling agents using a simpler amino acid structure that you can see here. And of course, the final outline should be like this, as it is shown here. And now I would like to point your attention a bit towards analytics of these kind of couplings. So my message is just, it's a bit tricky to confirm covalent bonds in a polymer backbone. And what is frequently used and was also used by us is HMBC, NMR, which practically tells you the correlation over several bonds between the substituent and the polymer backbone. And in our case, we think that we successfully confirmed the binding of this tryptophan residue by confirming correlations between the carbon 8 and the 9B and the carbonyl 10B of the substituent. So covalent bond confirmation is maybe NMR is the only real method that can do it, I'd say. Then finally, regarding these products, I mentioned charge titration at the beginning before. I said that a reduction of charge will tell you that something bound to the polymer backbone. And there's something that we found that's maybe interesting also for you. So you can, of course, produce all polysaccharide derivatives containing tryptophan with this substituent that I said already. So what's interesting, during charge titration, you can confirm also the stability of this methyl ester group if you want to attach something more or some longer peptides. Because during charge titration, you will also see that the methyl ester, of course, is labile. So at a certain pH of 9 or so, it will demethylate partially, and that's what you can see in the charge titration curves as well. So if you see here on the left side is plotted the carboxylic amounts of the polymer, and on the right side the free amino groups, and you see that in the product, say let's take this in the initial curve, so forward curve, which means from acidic to basic, the carboxylic acid amount is less than when you increase the pH, so you demethylate. So during charge titration something happens, of course, and important is that the amino groups are not visible in this charge titration, which means that the amide bond is still intact. So a very interesting way and simple way to quantify also the presence of your peptide on the polymer, as soon as you don't have too many functional groups, of course. And then the almost final example. So I promised you peptides, of course. So I was talking too much about small molecules, but here are the peptides. So proteoglycans, so you have the backbone of uronic acid, and you have some peptide attached to it from the amino end. And this is one of the examples that we made where we attached peptides with different length to uronic acid. So you see here, the simplest example, of course, is the glycine methyl ester. And the second simplest example, second most simple example, is the diglycine O-methyl ester, and then of course you can prolong it and play the game. In this case, an ethyl ester of the triglycine was used and coupled to the backbone. And the good thing about the uronic acid is, of course, that you have certain regioselectivity of the carboxylic group in the 6th position here, because there is only one every second unit, every second monosaccharide. So we think, at least from the analytics that we have so far, that it's really specifically binding to this C6 position, at least these 3 peptides or 2 peptides, and is not derivatizing the rest if you, under the prerequisite that you use the right coupling agent, of course. And one example or one possibility to confirm it, the presence is, of course, IR spectroscopy. So the right side shows the 3 peptides or pure peptides, which have a typical spectra, of course, having the amide and carbonyl bonds. And the left side shows the products of the different conjugates. But of course, no story or no research is complete without NMR. We could show with proton NMR, of course, the presence of the polysaccharide. In the uronic acid case, you have acetyl groups, so it's very nice, separated from the rest of the protons and the anomeric groups as well. And once you couple the simplest amino acid, you get, or you see, the presence of at least of the methyl groups easily. And of course, then if you play the game and use longer ones, you will also see the presence of the other protection groups. And especially what is nice if you use ethyl esters as protection groups for the carboxylic end of the peptide, it will be also nicely separated from the rest of the signals, allowing you to confirm the presence of it at least, not the covalent bond, of course. And the same you can do for carbon NMR. You will see that, of course, the longer the peptide becomes, the many more peaks you will get, especially in the carbonyl region, which is obvious. And of course, they are similar. So in general, of course, it's quite hard to identify which carbonyl shift belongs to which without correlation spectroscopy, which is clear. But combining the knowledge that you gain from, say, the presence confirmation from NMR and other things like titration or dialysis and stability, you will be able to at least confirm certain success of the synthesis. Good. Then the last example that I have regarding, because I was talking not only about proteoglycans, I was also talking about bacteria, so a very distant world in a way. I just would like to tell you that we also try a little bit to work on, say, modifying chitin and producing something like semi-artificial peptidoglycans. And you remember the image about the peptidoglycan dry layer from SEM. So it's a very thin sacculus and film. And we work a lot on thin coatings and films of materials. So we also try to a bit use this knowledge to generate these structures. And you know how, in the most ideal case, chitin looks like. And if you compare, of course, chitin, as I said already, with the peptidoglycans, the very simple task would be just to attach here a lactic acid residue and a peptide and cross-link the whole structure to end up with what you would like to have. And we are so far here that we can make thin coatings and we can derivatize it to a certain extent. So attaching, say, short amino acids to these kind of films is a relatively easy task, but of course, as you can imagine, chemically modifying it in solution, especially chitin, which is hardly soluble, is not the easiest task you can have. Good. Then I will come to the conclusion already, so that there is also maybe time for questions. So a conclusion and outlook about PPCs. What I wanted to say was that, of course, they are somehow inspired by existing biological structures. In our cases, it's proteoglycans and peptidoglycans. Of course, there are many more in nature, if you think about glycopeptide antibiotics and all these, but yeah, these are our main motivations. Definitely, PPCs have been prepared, so either attaching something to the chain or modifying the reducing end. This is, I think, our hope I could show. In any case, people use either coupling agents or oxidants to attach peptides to the structure. And that's maybe, if you're interested to work in that area, maybe a hint, I don't know, but enzymatic modifications are comparably less studied, I'd say. So if you work with enzymes, or if you have some enzyme toolbox to more specifically modify PPCs or create them, would be really interesting. Also, and I mentioned the work of Professor Edgar, lately published one, and there are of course others. Non-reducing end modifications are quite rare compared to all the other things that have been created. So of course, we're always looking for new stuff in science, if you like. And of course, the biggest challenge maybe reaches selectivity throughout the chain. I have not seen that unless you build the polysaccharide from the scratch, as Professor Seeberger and many other groups do. I don't see so far a method that would be able to do that. And finally, I also tried to think a bit about what is the most potential applications. And I mentioned immunology, so it's already used technically in a way. But of course, 3D printing and tissue engineering might be the most used. And drug delivery, which is somehow related to immunology, maybe could also be one that is interesting to study, but of course, you might have your own ideas. You know that we are going to have a conference soon, so the deadline for submissions is still in April, so please go ahead and submit if you haven't. And then finally, I would like to thank you for your patience, and again, thanks the organizers for giving the chance that I show some things, what we are doing. This is part of our group, and I also have to thank people from Slovenia that I mentioned here, who do a lot of NMR and surface analytical things for us. And yeah, we are looking forward to welcoming you in Graz at the EPNOE conference. Thanks a lot.